A very good morning to all. Today I'll be discussing about the topic protein sorting and transport. Protein sorting is actually the afterward process of central dogma. As you know that central dogma is the conversion of DNA into mRNA and then mRNA into protein. Means genetic code decodes it into mRNA, then the mRNA is converted into protein. Then this protein, where does this go? This protein goes to the final destination, and the process of sending the protein to its final destination, either it is in the cytoplasm, it is in the nucleus, it is in the cell membrane. This whole process comes under the category of, or you can say, and comes under the definition of protein sorting. So first I'll be discussing about the central dogma, DNA synthesis. It maintains, the first part of central dogma is DNA synthesis. It maintains the genetic information and passes this to the next generation. Then comes to the RNA synthesis. It is a transfer of the information from the DNA, where it is stored into RNA, which can be transported and interpreted. Then translation. It is the conversion of ribosomes, actually the use of ribosomes, which converts mRNA into amino acids. Amino acids finally forms a chain, a polypeptide chain, which is called a protein. Now that protein has been synthesized, now it will go to its final destination, and that destination is either nucleus, cytoplasm, cell membrane, or any other inclusion bodies. Now coming to the main part, that is protein sorting. Protein targeting, protein sorting, or actually these all are the same name, which is called protein sorting, and it is the ability of a cell to transport the protein to the appropriate positions in the cell or outside of it. But in prokaryotes and eukaryotes, newly synthesized proteins must be delivered to a specific subcellular location or exported from the cell for the correct activity. This whole phenomenon is called protein targeting, or you can say protein sorting. So the protein sorting is necessary for proteins that are destined to work outside the cytoplasm. The delivery process is carried out based on the information contained in the protein itself. Means you can say the address, means where this protein will go. It depends on the address which is written in the protein code. The correct sorting is crucial for the cell. Errors can lead to diseases. Now, coming to the discovery part. It was discovered by Gunther Blobel, who conducted experiments on the translocation of proteins across membranes. For this, he was awarded Nobel Prize. He discovered many proteins which have a signal sequence, that is a sort of amino acid sequence at one end, means that acts as address. You can say that signal sequence acts as address at one end, that function like a postal code for the target organelle, means that acts as a code. You can say the address to which part the protein will go, which is the destination of protein. That signal, that sequence is encoded in the protein itself. Now, what are the targeting pathways? There are two targeting pathways, that is post-translational translocation and co-translational translocation. Post-translational translocation is the pathway which occurs after the process of translation. Even though most proteins are co-translationally translocated, some are translated in the cytoplasm and later transported to their destination. This occurs for proteins that go to a mitochondrion, a chloroplast, or a peroxisome. Now coming to the co-translational translocation. In this pathway, transport of protein occurs during translation which is not completed fully. Synthesized protein is transferred to an SRP receptor. On the endoplasmic reticulum, a membrane enclosed organelle. Then there the nascent protein is inserted into the translocation complex. Now this This picture shows the overall pathways of protein sorting. Means either it will be translocated post-translationally or it will be translocated co-translationally. When it is translocated post-translationally, then it goes to mitochondrion, peroxisome, chloroplast, and nucleus. And when it is transported co-translationally, it goes to endoplasmic reticulum, Golgi apparatus, cell exterior, plasma membrane, lysomembrane, lysosome, and other parts of the endomembrane system. Now coming to the targeting signal, the postal code. The targeting signals are the pieces of information that enable the cellular transport machinery to correctly position a protein inside or outside the cell. This information is contained in the polypeptide chain or in the folded protein. In the absence of targeting signals, a protein will remain in the cytoplasm. So a continuous stretch of amino acid residues in the chain that enables targeting are called signal peptides or targeting peptides. There are two types of targeting peptides, the pre-sequences and the internal targeting peptides. Means some postal code will be present in the initial part of the sequence and some will be present internally at various positions. The pre-sequences of the targeting peptides are often found at the N-terminal extension. It is composed of between 6 to 136 basic and hydrophobic amino acids. In case of peroxisomes, the targeting sequence is on the C-terminal extension mostly. Signal sequences are removed from the finished protein by the specialized signal peptidases once the sorting process has been completed. And the internal targeting peptides are the peptides which are often found at or within the polypeptide chain, not at any end. It is not present at the end, it is present internally. Now this picture shows the position of signal sequences. In unfolded position proteins, it comes at N-terminal end. It is mostly found in N-terminal ends, and interspersed sequences are found within the whole protein sequences. Now, what are the pathways through which protein is translocated? Proteins can move between compartments in different ways. Like it moves through gated transport, like when it goes to nucleus. It moves through transmembrane transport, like mitochondria peroxisome, and then vesicular transport, like endoplasmic reticulum. Now, in gated transport, the protein traffic between the cytosol and nucleus, which occurs between topologically equivalent spaces which are in continuity through the nuclear pore complexes. The nuclear pore complexes function as you can say that nuclear pore function as a gate through which the protein passes. Now coming to transmembrane transport, some membrane bound proteins, translocators, translocators directly transport specific proteins across a membrane from the cytosol into a space that is topologically distant. The transported protein molecule usually must unfold to snake through the translocator. The initial transport of selected proteins from the cytosol into the ER lumen or from the cytosol into the mitochondria. Now coming to the vesicle transport, proteins from the ER to the Golgi apparatus and proteins to the ER, for example, occurs in this way. ER here means endoplasmic reticulum. Transport intermediates, which may be small, spherical, transport vesicles, or larger, irregularly shaped organelle fragments, ferry proteins from one compartment to another. The transfer of soluble is recognized by a complementary receptor in the appropriate membrane. So this is the whole introductory part of protein sorting. Thank you.